Hello guys, welcome back to our channel Biolab India, and this is Vamsi here at Biolab India. And in previous video of sexual reproduction and flowering plants, we have discussed till here, and we have seen a very introductory points on this lesson. In today's video, we'll be just moving a little deep. We'll be diving a little deep into the reproductive structures. And in today's video, we'll be seeing about the male reproductive structure that is called as your Androecium, male reproductive structure. Okay. Male reproductive structure. Generally, if you see a flower, I'm just drawing a simple flower here, a little flower, which is your basically a favorite flower of every teacher who is teaching biology or botany. That is your hibiscus rosa sinensis. That is the flower, China rose or anything, we call anything. I am drawing that flower here. This is the flower. Okay, I am just drawing you. I am showing you the outer thing. I am not going into the very deep inner thing. I will be showing you it a minute after, in a while. Okay. Generally, the flower looks like this to a human who is generally observing flower. A flower looks like this. Which contains two holes. What are holes? They are the rows in which different organs are arranged in a flower, like this. Hole, one hole, second hole. Here, if you see a flower generally like this, you see two holes. One, it is your petals, which are very colorful. Second one is your sepals, which are green in color. The function of these sepals is to protect the flower when it is in bud stage. These are colored. Okay, these are not sepals, very sorry. This is your sepal, and this is your thalamus. This is your stalk. Okay, like this is the general thing when you see the flower like this. But when you cut it, you'll be finding two extra holes which are very necessary. Two extra holes which I am drawing in black color. Just see, these are the two extra holes which you find. Okay, am I right or wrong? These are the two extra holes which you generally see when you cut. And this hole, this one is your male reproductive part, which is called as your Androecium generally, and in flowering plants we call it as stamen. And this role, which is your female reproductive part, which we generally call as gynoecium, and here in flowering plants we call it as carpel or pistil. Okay, carpel or pistil. Stamen, carpel, androecium, gynoecium, thalamus, stalk, sepals, petals. These are the different parts of a flower. In today's video, we will be discussing about this stamen. Stamen is a male reproductive structure which is generally called as Androecium. Okay, if you are very okay, I will be just removing this. Stamen. We will be discussing about your stamen. Androecium is stamen. In stamens, we have two different parts. One, it is your anthers, and second one is your filament. Anther and filament. In between these two, you have another thing called as connector. It is a tissue which connects anthers with the filament. Filament, it is just a stalk. It is just stalk. I'll be drawing you the diagram. Please watch. Please see. I'll be drawing you the diagram with this. Okay. This is a typical diagram of a stem. This part, which is generally lobe, this part is your anther, anthers, and this part is your filament, and this part is your connective. Okay, and this part, this portion is attached to the plant. This portion is attached to flower. In flower, it can be either attached to a petal or it can be also attached to thalamus. I have shown you the thalamus, the bulged portion from where these sepals, from where these petals, from where this stamen are attached, and they seem to be arising from that. Either it can be attached to a petal or a thalamus. Okay, and this end, which is very closer to the plant body, is called as the proximal end. Anytime if you come across the word proximal, I'm really sorry. Wait. Really sorry for that disturbance. And also for that wrong spelling. Please forgive me for this time. Proximal end. Whenever you come across this word proximal end, just remember that the end which is very closer to the body, which is closer to the body. Either it can be a plant body or a human body. Okay. And which is very opposite to this end is distal. Distal is the farther end from the plant body. There you find your anthers, and at the proximal end, this anther stamen is attached to either petal or thalamus. Never confuse that petal is a thalamus. Okay. This is the very thing that you have to see. Next one. Let us see the things that happen in anthers. Anthers are the place where your male gametophytes called pollen grains are produced. Generally, angiospermic anther it is bilobed, means contains two lobes as shown in here, as shown in the figure. It is dithecous, means it is made up of two theca. What are theca? Theca are nothing but layers. Okay. It is generally bilobed and dithecous. And each lobe, see, it is like this. If you just open the anther, if you just bust open the anther, it looks like this. And each lobe has two corners. It is four corners. It is four-sided. Okay. Each lobe, this is one lobe, this is the other lobe. Each lobe at its corners has got your microsporangia, where the original pollen grain formation takes place, where the original microspore formation takes place, and these are your microsporangia. Okay, microsporangia. Listen very carefully from this topic, since NCERT has confused. NCERT has written very confusingly, so that is why. Just listen with patience from here. Microsporangia are present at 4 corners. So there are 4 microsporangia in each anther, if it is a general angiospermic anther, and 2 microsporangia in each lobe. Okay, these microsporangia are covered with a layer or theca called as tapetum. That is the innermost layer. It helps in nourishment. It does not help in protection. It does not help in the dehiscence. Only the above 3 layers which are present above the tapetum, which you generally call as, which you name them as, yes, yeah, I'm just seeing whether I have missed any point. Yeah, which you generally call it as epidermis, endodermis, epidermis, endothecium, and middle layers. Epidermis, endothecium, and middle layers are the 3 layers which help in dehiscence of anther, which helps in protection of anther. But the last layer, which is the innermost layer, this helps in nourishment of your microsporangia and pollen grains. Okay, that is your tapetum. Now, in each microsporangia, at center, there is a group of cells called as sporogenous tissue. There is a very group of cells called as sporogenous tissue. Okay, I'll be drawing this in the next, after analyzing this. Each microsporangia at the center contains a group of cells which are clustered together, called as your sporogenous tissue. SP tissue means sporogenous tissue. And here all the thing happens. Everything happens only here only. Okay, these are homogeneous cells. These are not heterogeneous cells. They are not different cells. There are similar cells. Which form a cluster. In the middle of this microsporangia called as sporogenous tissue. And this is sporogenous tissue. Each cell in this sporogenous tissue is a MMC or each cell in this sporogenous tissue is a potential pollen mother cell. Each cell in the sporogenous tissue is a potential pollen mother cell or microspore mother cell. Each cell, remember my words, each cell, not the sporogenous tissue. Each cell in this sporogenous tissue is a potential, means it is a potential, it is having that potential to act as pollen mother cell and to give out microspores, which in return form pollen grains. Microspores or pollen grains are one and the same place. But the thing is, with simple upgradation, these microspores convert into pollen grains. These microspores convert into pollen. Both are one and the same. No worries. Each cell in this sporogenous tissue. A minute, guys. Let me check the time. Each cell in this sporogenous tissue is capable of producing microspores, and hence it is called as a mother cell. And microspores, it is microspore mother cells, and microspores and pollen grains are one and the same, so it is called as pollen mother cell. Each cell. Okay. Now while forming these pollen grains, these microspores, they are not formed as single, they are formed as a cluster of 4 cells. These microspores are formed as a cluster of 4 cells. Microspores are formed as a cluster of 4 microspores called microspore tetrad. Microspore tetrad. Okay. Microspore tetrad is nothing but it is a cluster of 4 cells of these microspores. Microspores are not formed in a single way, but they are formed as a microspore tetrad. Okay. And this process of formation of microspores from your PMC or MMC is termed as microsporogenesis. Okay. The process of formation of microspores from PMC or MMC is microsporogenesis. Microsporogenesis. Okay. Microsporogenesis. And this microsporogenesis, it is nothing but your reduction division or meiosis. Reduction division or meiosis. Here the NCERT asks you a question. That what is the ploidy of, where is that, what is the what is the ploidy of a tetrad? The ploidy of a tetrad is haploid. A tetrad, microspore tetrad, it is haploid. Microspore tetrad is haploid. And haploid. Okay, a microspore tetrad is a haploid. Clear? Microspore tetrad is haploid. Okay. Now, before we have discussed that this tapetum, about the tapetum cells are generally multinucleate. Okay, tapetum cells are generally multinucleate, but here NCERT also asks you a question how they can be binucleate. Generally, see, tapetal cells, or generally in general cells in cell division, once the karyokinesis, once the nuclear division occurs, the cell has to divide, which is called as cytokinesis. But in tapetal cells, this cytokinesis does not happen. Only karyokinesis happens. So karyokinesis is not followed by cytokinesis, and hence you can see binucleate tapetal cells. Binucleate tapetal cells means the cells with two nuclei. Okay, the tapetal cells with two nuclei. Very clear? Very clear? PMC. Okay, we are discussing that. PMC converts into microspores, and these microspores form a cluster called as microspore tetrad, and this microspore tetrad is haploid, and this sporogenous tissue. What is PMC? It is each cell of a sporogenous tissue is called as PMC, and this PMC is your 2N. It is diploid. From diploid to haploid, since reproduction divisions called as meiotic divisions are occurring in the cells. Okay, very clear. Now I have told you three layers that help in dehiscence of anther and protection. What are those? Epidermis, endothecium, and your middle layers. They help in dehiscence as well as protection. Now each anther, as the anther dehydrates, so as the water in the anther evaporates, as the water in the anther, I can erase this. Can I erase this? Yes. As the water in the anther evaporates or dehydrates, these microspores are converted into pollen grains, and after dehydration, complete dehydration, dehiscence occurs. Okay, dehiscence occurs as the anther dehydrates each microspore is converted into a pollen grain. Okay, a pollen grain. So a cluster of cells is called as a cluster of cells at the middle of your microsporangium is called as sporogenous tissue, and each cell in that sporogenous tissue acts as a pollen mother cell and forms each cell forms microspore tetrads. So each cell is forming 4 microspores. Okay, are you getting? Each cell in the sporogenous tissue is forming 4 microspores. Like that, there are many cells in the sporogenous tissue, and each cell is forming 4 microspores. If there are say for 20 cells in the sporogenous tissue, then how many microspores can be formed? 4 into 20, how much? 80 microspores can be formed. So like that, there are many pollen grains formed in this anther, and as the anther dehydrates and dehisces, these pollen grains are released into the air. These pollen grains are released into the air. Now we will be seeing the structure of pollen grains. Put the side heading, structure of pollen grains. Okay, video is getting little lengthy, no problem. These pollen grains are your male gametophytes. Okay, let us keep this video till here, no problem. Let us keep this video till here. In the next video, we will be discussing about your structure of pollen grains, guys. Okay, have a great day further, guys. Thanks for watching, and if you like this video, if you have learned something from this video, if your doubts, when you have read the NCERT, you have got many doubts that PMC undergoes meiosis and forms microspores and blah 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 blah, and they have written that each cell undergoes meiosis and forms PMC, and then PMC forms microspore tetrad, and microspore tetrad again undergoes meiosis. No, nothing. Microspore tetrad is formed. Okay, microspore tetrad is formed. Microspores, single microspore is not formed. They're formed as a cluster of flower cells, which is called as microspore tetrad. And I hope these small doubts and confusions have been cleared in this video for you. If not, if you have any doubt, please do not hesitate to mention in the comment section below. I'll be discussing it in the next video. This is the end of this video. In next video, we'll be discussing about the structure of pollen grain, guys. Thank you. Thanks for watching. Have a great day further. This is Vamsi signing off from Biolab India. Thank you, guys.